Hello students, welcome to online biology classes. So today's topic is double fertilization. So before this one, whatever the topic we covered, just correct that. So uh, we covered the outbreeding devices, that is the contrivances of the cross pollination. After that one, pollen pistil interaction we covered. So the small topic is linked to the pollen pistil interaction, artificial hybridization also we covered. So now double fertilization is topic. Before that one, how the pollen tube means pollen germination and pollen tube entry into the ovary. So that we have to cover in short it here. So what happens? Uh, uh, this is the stigma and the ovary. Yes. So pollination occurs. Now the right type of pollen grain on the stigmatic surface. So if it is a right type, the pistil will recognize this pollen grain and it will accept and promote the post-pollination event. Under that one, pollen germination and pollen tube growth through the stigma style and entry of the pollen tube into the ovary takes place. So now what happens? Pollen germination takes place. So through the germ pore, pollen tube produce one. That pollen tube gradually passes through the stigma style and almost come to the ovary. Clear? Now further movement of pollen tube show the some variations. What happens? So this is the anatropous ovule. Yes, inside to that one embryo sac is present. This is a female embryo sac. So the entry of the pollen tube into this ovule are three types. One based on the point of entry. The first one, if this is an anatropous ovule, yes, and this is the embryo sac. So the pollen tube will come and gradually it move towards the micropylar end. So the entry of the pollen tube into the ovary, if it is a toward the micropylar end, this type of entry of pollen tube is called the porogamy. Yes, so in this pollen tube, two male gamete and vegetarian nucleus is there. If pollen tube enter into the ovule through the micropylar end, so that is called the porogamy. Second type, this is again anatropous ovule. These are the integument, and this is the embryo sac. Yes, so this is the embryo sac, and this is also embryo sac. Second option, the pollen tube come up to the ovary and it will enter the ovule through the chalazal end only. Clear? And gradually and it will move towards the micropylar end. So here the entry of the pollen tube into the ovule through the chalazal end. So this type of entry is called as a chalazogamy. Yes, one more type is there. So if it is a anatropous ovule and these are the integument and this is the embryo sac. Now the pollen tube come from the stylar region and it will enter the ovule through towards the integument. Clear? And again reach towards the micropylar end only. But the entry of the pollen tube is always from the integument one. This type of entry is called the mesogamy. Yes. So ultimately, whatever the point of entry, either it is from the micropylar end or from the integument or the chalazion end, ultimately the pollen tube will reach toward the micropylar end only. Clear? So this is the entry of the pollen tube into the ovule. Now, what happens? This is an anatropous ovule. And this is the embryo sac. And this is the nuclear tissue. In embryo sac, 3 cells are present toward the chalazion end. These are called antipodal cell. Toward the micropylar end, 2 synergids are there. These are the synergids. In the center of this one, egg is there. Yes. In the center of the cell, again 2 polar nucleus are present. So this is a 7-celled 8 nucleus embryo sac present in the anatropous ovule. So when the pollen tube come toward the micropylar end, clear? It will enter through the micropylar end only and move towards the one of the synergid. Clear? This is a synergid, and this filiform apparatus, this black color portion is visible. This is called filiform apparatus. So this filiform apparatus provide the guidance, means it will direct the pollen tube towards the egg one. So this will assist the entry of the pollen tube toward the micropylar end. So once it enter toward the embryo sac, so one of the synergid degenerate and it will release its cytoplasma into the embryo sac only. So this pollen tube will come toward the micropylar end and towards one synergid and release its two gamete. Clear? So this is the entry of pollen tube into the ovule, always from the micropylar end only. So what will happen in the next one? That is nothing but the double fertilization. So before that one, you must know the type of entry of the pollen tube into the ovule one. Either it is a orogamy from the micropylar end, either it is a mesogamy from the integument, either it is a chalazogamy from the chalazion end one. Once the pollen tube comes toward the micropylar end, it always moves toward the one of the synergid. 2 synergids are there. It moves toward the one synergid. The filiform apparatus present inside the synergid, it will guide the pollen tube towards the embryo sac. Clear? So the entry of the pollen tube result into the degeneration of this, means rupturing of this synergid cell, and all the cytoplasma released into the embryo sac. So these 2 male gametes are released into the cytoplasma of this ruptured synergid. Now the next event is a double fertilization. What happens to the double fertilization? We will cover on this board only. Clear? With a short pause. Yes, students. So what happens? The pollen germination takes place on the stigmatic surface. Pollen tube come out through the one of the germ pore. So germination on the stigmatic surface. The same pollen tube show the growth through the stigmatic and the stylar region. So it will gradually grow towards the ovary region. Here. So as the pollen tube enter the ovary, it will move toward the micropylar end. The entry of the pollen tube from the micropylar end through the filiform apparatus of one of the synergid. Clear? The filiform apparatus, it will guide the entry of the pollen tube from this region only. Clear? Next one of the synergid degenerate and release its cytoplasma into this embryo sac only. So in that area only, whatever the 2 gametes present in this pollen tube, the pollen tube enter and this will release the 2 male gametes in this region only. Now what double fertilization means? So this is embryo sac. It contains 3 antipodal cells toward the micropylar chalazion end. This is chalazal end, and these are the antipodal cells. The center cell contains two polar nucleus, and toward the micropolar end, one large egg is there, and this is a synergid which is still active one, and this is a degenerated synergid, and pollen tube entered here. This is the pollen tube, and this will release the two male gamete into the cytoplasm of this degenerated synergid. Yes. Now the two male gametes are present in the embryo sac. Now what happens? One of the male gametes, these are the polar nuclei, this is egg, and this is filiform apparatus, and this one is a synergid, and these are the male gametes. Now what happens? So one of the male gamete move towards this polar nucleus which is present in the center cell and fuse with this one and result into the triploid cell, that is a primary endosperm nucleus. Clear? Another male gamete move towards the egg and fertilize with this one. So here two events take place. This is the first event, this is the second event. Now what happens? So this is male gamete fused with egg, this one, and this results into diploid zygote. Male gamete is a haploid, egg is also haploid, the resulting zygote is a diploid. This first event is nothing but the syngamy or generative fertilization. This is actual fertilization where the one male gamete fused with the egg and form the diploid zygote. This diploid zygote in future developed into the embryo. The second event, this is the first, second event, another male gamete here fused with the two polar nucleus. The central cell contains two polar nucleus, but before the just fertilization, these two polar nucleus fuse to form secondary nucleus. Means these are N plus N. So these are the polar nuclei. But just before the fertilization, both the polar nucleus fuse and form the second nucleus. So this haploid male gamete fused with this secondary nucleus, which is diploid one, and result into the formation of triploid primary endosperm nucleus. So this is a triploid in nature. Can you differentiate? So this event is called as a triple fusion. Now, the pollen tube enter into the embryo sac towards the one of the synergid with the help of the filiform apparatus. So the synergid will degenerate and release its cytoplasma into the embryo sac. So in that cytoplasma only this pollen tube releases the two male gamete. Now out of these two male gamete, one male gamete move towards the egg. This male gamete is haploid and the egg is also haploid. So they fuse to form the diploid zygote. That actual event is called as the syngamy or generative fertilization. This is the actual means this development to the embryo one. So this is first event. Second event is another male gamete move toward the center cell where the two polar nucleus are there. So before the union of these three nucleus, these two polar nucleus fuse to form the secondary nucleus, which is a diploid in nature. That diploid secondary nucleus and the male gamete fuse to form triploid primary endosperm nucleus. So after fertilization, the embryo sac, now how it appears. So this is the embryo, same embryo sac. So towards the chalazal end, three antipodal cells are there. So these are degenerated ones. Clear? Towards the micropylar end, this is the zygote, which is a diploid in nature, and one synergid is degenerated, and this is another synergid. This will also undergo degeneration. Here, so this is a synergid. In the center, this is a primary endosperm nucleus. So this is the fertilized embryo sac. These are the degenerated antipodal cell. This is central cell. This is a primary endosperm cell, and this nucleus is a primary endosperm nucleus. Yeah, so short form. And this is zygote, which is diploid in nature. The primary endosperm nucleus is a triploid in nature, and this is synergy. So what is the fate of this zygote and the primary endosperm cell one? So this zygote in future develop into embryo, yes, and this primary endosperm cell, this will develop into endosperm. So this embryo and the endosperms are the post-fertilization structure. So actually now we are going to define this event. Double fertilization means it is a unique feature seen only in the angiosperm flowering plant. So double fertilization means there are two events when the syngamy and triple fusion which will take place in the single embryo sac. That complete event is called as a double fertilization. So first event is called as a syngamy, second event is called as a triple fusion. So this is a double fertilization. Sometimes it is also called as a double fertilization triple fusion. Here, so we are going to write the definition here. So with a short pause. Now the definition of the double fertilization. It is a unique phenomena seen only in angiosperms when synamine and triple fusion take place in one embryo sac, then that process is called as double fertilization. So actually two fusion take place. So after double fertilization, zygote develops into embryo and primary endosperm cell develops into endosperm. Yes. So double fertilization means it is a unique phenomenon seen in the angiosperm where the two fusion take place in the single embryo sac. First is the synamine where the male gamete fuse with the egg and develop into zygote. That zygote in future develop into the embryo. Second event is called as a triple fusion where the male gamete fuse with the polar nucleus. So here the three haploid nucleus fuse and form the triploid primary endosperm nucleus which is present in the center cell. That is after double fertilization, it is called the primary endosperm cell. So this primary endosperm cell develop into the endosperm. So the embryo and the endosperm are the post-fertilization structure. So this is about the double fertilization. So next heading is the post-fertilization structure and the events. Clear? So today we are going to cover only the endosperm formation. Next, after that one, embryo formation is there. Clear? So just wait for 2 seconds. So next heading is the post-fertilization structures and the event. So after fertilization, what happen in this flower or the ovary? That is the main topic. One post-fertilization event and structure means, so after double fertilization, the development of endosperm embryo and maturation of ovules into seeds and ovary into fruit is collectively called as post fertilization events. So what happens? So after double fertilization, which will take place in the embryo sac, the endosperm develop from the primary endosperm nuclear cell, the embryo develop from the zygote. So these two structures are formed, the endosperm development and embryo, followed by the transformation or the maturation of the ovules into seed. The complete anatropous ovule turn into seed one, and the ovary which is developed into fruit. That complete process is called the post-fertilization event. So in that one, the post-fertilization structures means the endosperm is the first, the embryo is the second, then the seeds are the post-fertilization structure, and even the fruit is also post-fertilization structure. Clear? So under that one, grad means one by one we are going to cover the embryo, endosperm, seeds and the fruit one. So this is about the post-fertilization structure or the event. The first heading we are going to cover, that is the endosperm. Yes, so endosperm. So this endosperm develops from primary endosperm cell. So in short, it is also called the primary endosperm cell. But the embryo development take place only after endosperm formation. So try to understand this one. So in textbook what they given, the endosperm formation precedes the embryo formation. So until and unless endosperm is not formed, embryo development will not take place. That's why embryo development take place only after the endosperm formation. Now how the endosperm development take place? So this is the embryo sac. These are the degenerated antipodal cell, and this is a diploid zygote, and this is the primary endosperm nucleus, and this cell is called the primary endosperm cell. This zygote develop into embryo, and the center cell, now I will take this center cell, is called as the primary endosperm cell with the primary endosperm nucleus. This is the primary endosperm cell. Based on the pattern of division, the endosperms are the briefly three types. Clear? So if this endosperm division, three types. First one, if this endosperm nucleus undergo repeated nuclear division after, clear, without followed by the cell wall formation, means this primary endosperm nucleus undergo repeated mitotic division and result into the primary endosperm cell, where the millions of primary endosperm nucleus. This repeated nucleus division takes place. Clear? Means here karyokinesis is not followed by cytokinesis. That's why this primary endosperm cell contains only nucleus in that cytoplasma. This type of endosperm is called as a free nuclear endosperm. Clear? Free nuclear endosperm. Second type, if this primary endosperm cell undergo karyokinesis followed by the cell wall formation, after every nuclear division, cell wall formation takes place, and ultimately endosperm look like this one. After every division, cell wall is formed. Here, karyokinesis followed by the cytokinesis. Kinesis followed by cytokinesis. This type of endosperm is nothing but cellular endosperm. This is the second type. Can you differentiate? If the primary endosperm nucleus undergoes repeated nuclear division not followed by the cytokinesis, it contains only free nuclei. Clear? So this is called the free nuclear endosperm. Second one, if the primary endosperm nucleus undergo division followed by the cytokinesis, for after every cell division, cell wall is formed, nuclear division, cell wall is formed, that is called the cellular endosperm. In between that one more hybrid type is there, that is called as a, if the primary endosperm nucleus undergo first nuclear division followed by the cell wall formation, but this is micropylar end and this is chalaza end. Only during the first division cell wall is formed. After that one, further division is also again free nuclear division. So this is micropylar end where the many nuclei are present in the cytoplasma, and this is the halobial chalaza end. This also contains the free nuclei. So this type of his endosperm is called as a halobial endosperm. So in NCERT textbook, the type of endosperm is not mentioned. So cellular endosperm, free nuclear endosperm. When you take the coconut one, so this is a coconut. So initially it contains only the, if it is a tender coconut, it contains only water. In that one only nuclei are present. Clear? So initial stage of coconut, if contain only water, then it is called the free nuclear endosperm. Gradually, as it matures, so the kernel, white kernel will develop towards the outer cell. Yes, gradually water quantity decreases, so the kernel which is present toward the periphery, so this is a cellular endosperm, which is nothing but the kernel. This is a nuclear endosperm. which is nothing but the water. Yes. So during the maturity, the nuclear endosperm gradually turn into the cellular endosperm. So this is the coconut case. Yeah. So whether this endosperm is, what is the function of endosperm? So the main function of endosperm, the endosperm provide nourishment to developing embryo. That's why the endosperm formation is a very important. Until and unless endosperm is not formation, embryo development will not take place. So that's why it will act as a nourishing tissue. So the major question, whether the endosperm is completely utilized or not. Here, so when you take the mature seeds, so some seeds are there where the endosperm is still present. In some seeds, endosperm is you can't get. Here, for example, when you take the peas, beans, and groundnut. Clear? In these three, endosperm is completely utilized by the developing embryo. Yes. So what it means, the embryo will consume the endosperm completely. That's why in mature seed, endosperm is missing. When you take the peas, beans and groundnut, the endosperm is completely consumed. Clear? So one more example is given, that is the castor and maize. So in this case, the endosperm is partially utilized. Due to that one, in mature seed of the castor and maize, they contain the small amount of endosperm. Based on the presence or the absence of endosperm, the seed classification is carried out. So this is about the endosperm. The endosperm developed from the primary endosperm cell. Based on the nuclear division or the type of division, the endosperms are either free nuclear endosperm or the cellular endosperm. Halobial endosperm in between this one. It is the intermediate type. So this will show the both character. Here. So endosperm will act as a nourishing tissue for the developing embryo. So during the embryo development, the endosperm is either completely utilized or the partially utilized one. When you take the groundnut, pea and beans, the endosperm is completely utilized. That's why in mature seed, endosperm is totally absent. When you take the castor and the maize, the endosperm is a partially utilized. That's why these seeds contain the endosperm. So in next video, we are going to cover the embryo development. In that one, monocot and dicot embryo is totally different. After that one, seed is there, then the fruit and the remaining topic. So we'll meet in the next video. Till then, take care of yourself. Till then, goodbye.